All this is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. So, as I continue to work on my book, there is a gap in my video lectures. However, this is a study that came out of China. This study talks about the autoimmunity or autoimmune attack against the brain tissue in long COVID patients. The results of that is the patients have fatigue, they have psychological issues, depression, they have brain fog, cognitive decline, and so on. The basic problem in this study that they found was that as the immune system attacks the brain tissue, the important aspect is the blood brain barrier that must be compromised as well. The reason is very simple. They saw antibodies attacking the neuron and the various proteins and enzymes in the myelin sheath or in the neuron, which I would discuss in a second. However, for an antibody to go from the body, the systemic circulation, into the brain means that it needs a way to enter there. Normally, in a healthy individual with healthy blood-brain barrier, the antibodies are not, it's not easy for these antibodies to enter the brain tissue. Yes, there is some carriage of an antibody from the vascular side into the brain, or there are special transporters that can transport some antibodies as well, which are present in the blood-brain barrier, but they are negligible. So systemic inflammation should not, through antibodies, should not attack the brain. However, if the antibodies are found in the brain, that means the blood-brain barrier is compromised. So when you as a physician or you as a patient are talking with your clinician, the approach towards the neuroinflammation is not only to reduce inflammation, but also to stabilize the blood-brain barrier before the antibodies and inflammation in the brain can actually be taken care of. So that is the basic summary of the study. Let me start with number one. I'll show you the study, then I would show you the mechanism and the findings of the study. So I hope audio is fine. So these are the gifts for humanity, and they are continuing, especially for long COVID and individuals with the vaccine side effects. This is the neuron, and look at these diagrams that I made for you for this lecture. Actually, I'm doing diagrams for my book as well, so that is how these diagrams are coming together. This is the study published in Science Direct, published on November 9, 2024. The study is brain targeted. Autoimmunity is strongly associated with long COVID and its chronic fatigue syndrome, as well as its affective symptoms. So here they actually coined a new term, affective symptoms. In the affective symptoms, what they mean is physiological symptoms, that is fatigue, and psychological symptoms, for example, depression and other such symptoms. So together they're calling them affective symptoms. So let's look at the highlights. They say brain targeted autoimmunity is key in long COVID pathogenesis. And this is very important for diagnosis. They are saying immunoglobulin, M and G against myelin basic protein. And I would explain what is myelin basic protein, or MBP, and myelin oligodendrocyte glycoprotein. I'll explain that as well, what is MOG, predict long COVID. So antibodies against these two proteins predict long COVID. Then they say IgM against synapsin. Again, I'll explain it. IgA against MBP, IgG against MOG, and C-reactive protein, which is a inflammatory biomarker, protein predict effective symptoms in long COVID. So what is interesting, one more interesting thing in this study that you might actually just read, by the way, in the study, is that the peak body temperature at the time of acute COVID, meaning when somebody was ill with COVID, then what was their body temperature, the peak that was attained? If the peak was high, then there is a chance for having a severer long COVID. And the reason is simple: peak temperature means an intense immune response, and the temperature or the fever occurs as part of the immune response. So, higher the temperature means more intense the immune response, which then means also a possibility of autoreactivity or autoimmunity or long COVID. So, I want to read the abstract before going to my diagrams and explaining them. So, they say autoimmune response contributes to the pathophysiology of long COVID. Affective symptoms and myalgic encephalitis or encephalomyelitis, chronic fatigue syndrome. So they actually, interestingly, also compared the immune profile of ME/CFS patients, myeloencephalitis, chronic fatigue syndrome patients, whose symptoms look very similar to long COVID neurological patients. So they said we wanted to examine whether long COVID and its accompanying affective symptoms and CFS, chronic fatigue syndrome, are associated with immunoglobulin A, M, G directed at neuronal proteins, including myelin basic protein, oligodendrocyte glycoprotein, synapsin, alpha and plus beta tubulin, neurofilament protein, cerebellar protein, and the blood-brain barrier damage protein or Claudin 5 and S100B. And I'll show you all of those proteins in my diagrams in a second. I just want us to kind of look at the study. So they had a method, I'm going to leave that, other than 90 long COVID patients and 90 healthy controls were used. And please remember, C-reactive protein is a protein that is produced by the liver when we have an acute infection or inflammation. There are messages that are sent to liver to say we need more proteins to fight inflammation. And in response, liver produces a number of proteins. Those proteins are called acute phase reactants. They are reactants, proteins. They are produced for the acute phase of an infection or inflammation. C-reactive protein is actually an acute phase reactant. It is not only for biomarker. Liver doesn't produce it only for the labs to be used, but actually it goes in the inflammatory area and works there, especially with the complement system, and helps in inflammation. The second is oxidation protein products. This is also very important, AOPP. Oxidation protein products or advanced oxidation protein products, these are the products or the proteins that are produced when the reactive oxygen species are high in inflammation, and these species are attacking various proteins, misfolding them, damaging them, and now those proteins are seen in the blood circulation as well. So these two, the CRP and AOPP, are indicators of inflammation in the body. And then the antibodies against neurological tissue are indications of the brain tissue being attacked. So here are the results. I'm going to explain these with my diagrams, but generally what they're saying is long COVID is associated. Let me see if I can make it a tiny bit bigger. Long COVID is associated with significant increase in IgG directed at tubulin. Tubulin is a structural protein within the neurons. MBP, myelin basic protein, which is also wrapping around the neurons, and it is a protein in that wrapping. MOG, or myelin oligodendrocyte glycoprotein, and synapsin, that is a protein that is present in the synapse of the neuron. All of that with the diagrams a little later. IgM, MBP, MOG, CP2, synapsin, and BBD, and IgA, CPT, CP2, synapsin. So they're saying various kinds of antibodies are seen, observed in long COVID patient, statistically significantly different from healthy individuals. They also saw something very interesting. IgM against SARS-CoV-2 in some patients, which means IgM's presence means an acute problem, a present problem. So IgM against SARS-CoV-2 means probably SARS-CoV-2 is still hiding somewhere. IgM against HHV6, human herpesvirus 6, so possibly a reactivation. So these antibody titers were significantly correlated with IgA, IgG, IgM tubulin, and CP2, IgG, IgM, BBD, IgM MOG, IgA, IgM NFP, and synapsin. So not only there was brain tissue under attack, there was possibly persistent virus and possibly reactivation of herpesvirus. And they actually talk a little later that even EBV virus. So they say binary logistical regression analysis shows that IgM MBP and IgG MBP are the best predictors for long COVID. Again, small study, 90 people, but still very useful. So if somebody has long COVID and if they have neurological symptoms, the basic test that can be done according to this study is the presence of IgM against MBP and IgG against MBP. Multiple regression analysis, so multiple variables were seen, shows that IgG, so the difference between IgM and IgG, as you all know, IgG is for a longer period of time. When we have an acute attack of some virus, some bacteria, some fungus, or some inflammatory state, IgM is produced first of all. Once the IgM is produced and inflammation is now, being curtailed, then the body would start producing IgG that can continue to go on for 3-4 months or even longer. So IgG kind of stands guard. If the same event occurs again, it is present and it is helping. Also remember, IgG can cross placenta and go to baby as well to provide protection. So they're saying multiple regression analysis shows that IgG, MOG, CRP, and AOPP, remember these CRP and AOPP are the inflammatory state and reactive oxygen species state, together 41.7% of the variance in the severity of CFS. So the severity of chronic fatigue syndrome, how severe it is or how less it is, these three markers were correlated with the severity. Neural network analysis shows that IgM synapsin, IgA MBP, IgG MOG, and IgA synapsin, IgA CP2, MBP, and CRP are the most important predictors of affective symptoms due to long COVID with a predictive accuracy of 0.8. This is also very important to keep in mind. So these are the labs that can possibly be done if somebody has not only neurological symptoms, but also psychological symptoms. So that is what they're calling combined affective. Conclusion: brain-targeted autoimmunity contributes significantly to the pathogenesis of long COVID and the severity of its physio-affective phenomena. I want to show this diagram from them. I really liked it. And in this diagram, what they're saying is, if you see from top to bottom, long COVID and chronic fatigue syndrome patients, they examined their serums, IgA level, IgM, G to CNS proteins, herpesvirus proteins, and SARS-CoV-2. What they found was, this is interesting. So if you wanted to have an idea of what labs and what is raised, here it is. IgA against CP2, we'll discuss that. CP2 stands for cerebellar protein type 2. And synapsin. IgM against MBP, MOG, CP2, and synapsin. That is an acute effect, IgM. And then IgG against tubulin, MBP, MOG, and synapsin. Then they say best predictors for long COVID disease, especially neurological, based on this study, IgM against MBP and IgG against MBP. Then they say for CFS, chronic fatigue syndrome patients, not necessarily long COVID, IgG against MOG, and CRP and AOPP, and then, as you saw, the rest as well. So this one diagram is really good diagram. Now, the study is long and thorough, and there is a lot to be seen in here, especially this table here is also very interesting, where they give various antibody levels and their significance. This table number 2, is also very, very interesting. However, with this study in front of us, I wanted to quickly go over with my diagrams for what they saw. The diagrams that you're going to see are really the diagrams that I'm drawing for the book I'm writing. The book should be out soon. So here is a neuron. A neuron in the brain has a neuronal body that is a cell. So cell body is present. There's nucleus or mitochondria, other things here. Neurons have dendrites with which they connect with the other neurons. Then they have a long process, usually, mostly just one long process, which is called axon. Through this axon, the electrical activity from the neuron gets out towards other neurons or other effector tissues like endocrine tissue or muscle tissue, etc. The axon is covered by a sheath, a fatty sheet. It is actually a cell that is called oligodendrocyte, and I'll show that in a second. That oligodendrocyte, that cell, creates a wrapping around the axon, like an insulation material. But it is not only for insulation, it also helps the nerve conduction or the impulse in the nerve to travel from one node of Ramvirr. Node of Ramvirr is the gap between this wrapping, from one node of Ramvirr to the next. And this is called the saltatory conduction or fast conduction. And this is how the neurons can send messages with lightning speed. Then there is, of course, a synapse, or sorry, I'm calling it synapse, nerve terminal. Inside the nerve terminal are many vesicles that are filled with neurotransmitters. Those neurotransmitters could be sent here from the cell body above, from here, or they can be manufactured here, or they can be picked up from the environment. So many reasons, and that can be a long discussion. However, whenever the electrical impulse reaches the nerve ending, then the nerve ending will release the neurotransmitter. If you see here, these little structures that I've made, and they all are useful, that is why I'm discussing them. This little structure that I've made here, the thread-like things, they are synapsins. These are proteins that are present in the pre-synaptic nerve terminal. Pre-synaptic means that the nerve terminal of a neuron that is coming and will release neurotransmitter on another neuron or effector tissue. So these structures are present in here, and they help release neurotransmitter. Think about it for a second. If for some reason synapsins aren't working correctly, then the neurotransmission will become a problem. And if neurotransmission becomes a problem, if we cannot release neurotransmitters, then even if the neuron is working correctly, its effect will not appear because neurotransmitters are not released. Does this make sense? So keeping an eye on all these parts of the neuron, because you'll see antibodies against the cell body, you'll see them against the tubulin inside the axons, against the myelin sheath, and against the synapsin. And I'll pose this question to pique your curiosity once more. Why will we have antibodies inside the brain? Antibodies are not allowed in there in general. So until the blood-brain barrier is compromised, until there is systemic inflammation, which is also causing inflammation of blood-brain barrier, these antibodies will not reach in the brain. So there has to be a continuous inflammatory state, which then compounds the problem by making the blood-brain barrier permeable and allowing these antibodies to enter the brain tissue. So here I have the next diagram, this is an oligodendrocyte. So this is a cell. This cell manufactures a lot of fatty material. That fatty material flows through small dendrites. That is why it is called oligodendrocyte. The dendrites are these small branches. It flows through the small dendrites. These dendrites then become expanded at the end, and then they wrap around the neurons. So if I go back here in this diagram, there will be an oligodendrocyte over here, and it would be sending in the dendrites to these areas where the wrapping is happening. And these wrappings, the myelin wrapping, are actually end pods of the oligodendrocytes. Now, inside the oligodendrocyte, there is another protein inside of it, inside the cytoplasm, inside the cell. There's a protein called myelin basic protein. Myelin basic protein does its own functions within the oligodendrocyte, so we are not concerned with the function of it, but we want to know that there is a protein inside the oligodendrocytes called myelin basic proteins, or MBP. On the outer side of the myelin cells or the oligodendrocytes, there is another protein called myelin oligodendrocyte glycoprotein, or MOG, MOG. And you just read that in the study that there were antibodies against MOG. So there are going to be antibodies against MOG, and there are going to be antibodies against MBP. The question is, MBP is hidden inside a cell. Antibodies don't go inside the cells. So how come we have antibodies against a proteinaceous structure that is sitting inside the cell? The answer is going to be very simple. Until we cause damage to the cell, which causes the MBP to spill out, and that spilled out MBP is then looked at by the immune system, and immune system then reacts against it. That is how the immune system will become primed. So tissue damage is an important first event to occur for these internal proteins to become a target of the immune system. So now here I'm discussing, or I'm showing, how the damage causes the internal structures of a cell to become exposed. So imagine that this is a neuron's cell body. An inflammatory system caused damage to the cell body, for example, by reactive oxygen species and other inflammatory molecules. When the damage occurs and the neuron breaks down, the neuronal structures that were hidden inside the cell will spill out. They will become exposed to the immune system. Now, interestingly, normally our immune system knows us. We are self for it, and it does not attack self. However, those proteins that are hidden inside the cells, when they are exposed to immune system can become mistaken and think of these proteins as foreign material and start attacking them. Another reason is that when these proteins are exposed, the proteins that are sitting inside the neuron, for example, or any other cell, when they are exposed to reactive oxygen species would cause deshaping, misfolding, and damage to these proteins. So when these proteins come out of a broken cell, they're not only in their healthy state, they're actually all mangled up. And so now immune system can make a mistake again, thinking they are foreign components and start attacking them. And in that process, making antibodies that would attack normal, healthy neurons as well. So same is the problem with the myelin sheath as well. When there is damage to the myelin sheath, the oligodendrocyte, then the components of the myelin sheath, the myelin basic protein and the MOG, they would spill out. MOG is already out, and they would have a problem. Immune system would react to them. Similar issue is with the axon. So here, this tiny blue area that I'm showing is the axon. When the axon is damaged and it opens up, inside the axon, there are many structures, but one of them is tubulin, Tubulin, from the word tube, is these are structural proteins present within the axon to, number one, give shape to the neuron, and number two, allow a neuron to connect with the other neuron. So those tubulins, when they become exposed, they will become a target for the immune system as well. And finally, the last part of the neuron, the nerve terminal. This nerve terminal, when that gets damaged, of course, the inside structures of the nerve terminal will also become exposed. And an important structure here is the synapsin. These little thread-like structures I've made. Synapsins are busy with the neurotransmission. So now this neuron is a lost story. It is already damaged. However, when the synapsins are going to spill out and the immune system is going to look at them and start attacking synapsins of healthy neurons, then we have a problem. So this is why throughout my discussions for 5 years, I've been saying controlling inflammation is paramount, because inflammation not only causes tissue functional problems, but it breaks down the tissues, the cells, exposing their inner components to the immune system, causing immune system to become primed against our own tissues, producing an autoimmunity. Okay, so continuing. So then here is the sequence of events. The neuron got damaged, various kind of proteins in the cell body, in the myelin sheath, in the axon, in the synapse became exposed. So here is an example. So look at this oligodendrocyte, that happy little cell, and one of its pods is damaged by inflammation. And this was the pod, let's say, that was wrapping around over here. So when that got damaged, the myelin basic protein came out. And they write it in their manuscript. They say that the myelin basic protein will spill out of the oligodendrocytes. When it spills out, where does it spill? It spills within the brain tissue. And over there, it becomes antigenic, meaning it triggers the immune system to react to it. Not only that, the myelin basic protein will then stick to other neurons that are in the vicinity. So brain is not an empty cavity. For majority of us, it has neurons. So the neurons that are nearby will have myelin basic protein sticking on their outer surfaces. Why? Because it got spilled out of a broken neuron. So this myelin basic protein that is now sticking, so let me see if I can zoom in. So here is a myelin basic protein from another broken oligodendrocyte, maybe from here, is sticking here. One piece is sticking here, one is over here, one is sitting out in the interstitium. All of this would cause immune cells to start attacking healthy neurons, mistaking them for being foreign material. So once outside the myelin basic protein becomes antigenic, meaning triggers the immune system, activating immune responses and contributing to neuroinflammation. And finally, this is my last diagram. Finally, what then happens? What are the antibodies they saw? So here, just a quick note about the color coding. All the red are IgAs. These were the IgAs that were statistically significantly elevated for long COVID, for example. Then in orange are IgMs that they observed, and in blue are IgGs that they observed. So if I go from the top to bottom of a neuron, in the cell body of the neuron, the cerebellum protein or cerebellar protein 2, normally this protein is found abundantly in the neurons present in the cerebellum of our brain. However, this protein is found in other neurons as well. So they found IgA and IgM targeted against cerebellar proteins. So neuron as a whole is under attack. And if the cerebellar proteins are abundantly present in the cerebellum, then cerebellum can be under attack. The function of the cerebellum is gait is an important one. It does many other things. But gait is important, the walking, because cerebellum helps with the muscle movement coordination. So CP2 antibodies were found. Then if I go down here to the myelin area, they saw antibodies against MOG, myelin oligodendrocyte glycoprotein, and MBP as well. So which antibodies they found against MOG? They found IgG, but that was not, I believe, significant. And they found IgM, acute inflammation. So in this final diagram, what I'm going to present to you is the antibodies that they saw and found in long COVID patients, actually in the chronic fatigue patients as well. But of course, our target is long COVID. So here what they saw was within the neuronal cell body, they saw antibodies that were targeted against the CP2. CP2 are the cerebellar protein type 2. These proteins are usually found in the cerebellum of our brain tissue. However, these are found in other neurons as well. They're more abundant in cerebellum. So CP2 protein, there were IgG against it, and IgA and IgM were against these proteins. Then for the myelin sheath, they saw two proteins of the myelin that were under attack. One was the MOG, or myelin oligodendrocyte glycoprotein. That is a protein that is present on the outer surface of the myelin cells, and they were targeted by IgG and IgM. Of course, that would cause myelin disruption and damage, and neuronal function will become compromised. Then is the myelin basic protein, or MBP. This is a protein that is inside the myelin, as I showed you before. And there were IgG, which is immunoglobulin G, that is the long-term immunoglobulin attacking this sheath. And then IgM, which is acute. And they said that this was the best predictor of long COVID. So somebody has neurological symptoms, and they are suspecting that this may be long COVID. Then IgG and IgM against MBP. So these will be called anti-MBP IgG and anti-MBP IgM. These were the best predictors. Then they saw antibody against tubulin. So as I said before, tubulin is a structure that is present within the axons, and it provides not only structure, but also helps axon find other neurons and connect with them. So very important structure. Tubulin targeted by IgG, long-term, IgA, and IgM. And then they saw in the synapse, the synapsin was targeted by IgA, IgG, and IgM. So almost the neuron itself, the end, the nerve terminal of the neuron, the wrapping of the neuron, these were all being targeted. And then to the sadness, they also found, when I was reading the study, I said, all right, maybe the blood-brain barrier is okay, and that is why these antibodies will not enter the brain and they will not cause an issue there. However, they found in these long COVID patients the blood-brain barrier protein targeted as well. That means a patient's immune system is generating antibodies against their blood-brain barrier, which, when the blood-brain barrier is attacked, becomes inflamed and permeable. And then all the other entourage of other antibodies would then spill into the brain and then go and attack various brain tissue parts. So that is the study. Apologies for the disruption in the middle of it. I will edit that part out and it will become okay in another day or so. So I'm just going to quickly answer a couple of questions. Sigrid says, can they be found in body peripheral nerves? So this was a central nervous system study. Peripheral nerves have been shown to have antibodies attacking them as well. So that is small fiber neuropathy. And similarly, the autonomic nervous system gets attacked as well. The ganglions get attacked as well. So these are different studies. If you like, I can discuss those studies as well. This particular study was only looking at the brain central nervous system, and that is why their antibodies that they presented were there. And Son, I'm so sorry to hear about these presentations. I hope that some of these studies, nowadays I'm working on the neurological aspects of the long COVID, ME/CFS, and so I would share some more studies as well, and prayers for your health and so on. Um, so this is the discussion. Thank you very much. Please like, subscribe, and share. Nowadays, my channel's viewership is going down because I come online less often. So whenever you get a chance and you see one of my videos, just like and comment, and that would help. With this, thank you very much, and I would see you next week. Hopefully, have a nice, great weekend. And if you would like to support this work, there are links in the description. If you would like to have CMEs to these lectures, there you can go to drbean.com. Link is also in the description. And also the link to this study is also in the description of this video. Thank you and have a good night.